Hello, folks. Today we are going to talk about membrane transport. Learning outcomes for the day. Recognize that metabolism is compartmentalized within cells. We'll compare and contrast the mitochondrial inner and outer membranes. We will recognize that cytosolic or glycolytic NADH does not enter the mitochondrion, but its electrons are shuttled through the glycerophosphate shuttle onto the electron transport chain. And we'll discuss how the protein gradient is essential for the transport of molecules such as pyruvate, ADP, and ATP, and phosphate in and out of the mitochondrion. Our Meet a Scientist moment of the day is Hamid Haroun. He is a research associate at the University of Manchester and is a wheelchair user. When he was young, he thought he wanted to be a physician, but he was discouraged to pursue this because of his disability. So instead, as he was still kind of pursuing this interest in medicine, he discovered how the sciences can also be related to medicine, and he found himself actually more in love with that aspect of medicine, the science behind it. So he ended up getting degrees in both medical physics and a PhD in MRI analysis. And so now he develops scanning protocols to image the brain, particularly brain structural changes in the early stages of dementia, which is pretty cool stuff. So let's kind of take a step back for a moment. Within our cells, various metabolic processes are regulated through allosteric regulation, like we've talked about, but also an aspect of regulation happens through compartmentalization. So glycolysis is happening in the cytosol. And as we move on to the citric acid cycle, that's now happening in the mitochondrial matrix, a different space. And then, even slightly different, the mitochondrial inner membrane is going to house most of the complexes of the electron transport chain, and that's where ATP synthesis happens as well. And this kind of separation of metabolism is important for its regulation. So let's talk about that today. Specifically, let's talk about the mitochondrion. So on this slide, I have on the left an electron micrograph of mitochondrion that's showing you kind of the structure as it looks like in real life. And on the right is a cartoon. You can see some various aspects of this organelle, such as its double membraned nature, so it has both an outer and inner membrane. And the inner membrane, folds in on itself, giving us more surface area to work with. And within that inner membrane is the matrix. Between the inner and outer membrane, the inner membrane space. So these are all kind of good things to know and to think about and visualize as we talk about the various metabolic pathways. Let's compare the inner and outer membrane. The outer membrane of the mitochondrion is mostly permeable to small molecules. Anything can kind of pass in and out as long as it's fairly small. Because within the membrane are these very large protein complexes called porins. They are integral membrane proteins that span the membrane. They're like big holes. They allow diffusion of molecules up to 10 kilodaltons, which is rather large for a small molecule or a small peptide. And because of the permeability of this membrane, the inner membrane space is mostly like the cytosol. We will learn that's not entirely true. Very, very close to the inner mitochondrial membrane is where the proton gradient exists. So there can be local differences, but largely the inner membrane, like the cytosol. Now the inner membrane is very different, okay? The inner membrane is permeable only to two gases and water, oxygen, CO2, and water. That means almost nothing is passing through this membrane. So how do things get inside the mitochondria or out? Through proteins. The inner membrane is 75% protein by mass, and we have imaged this again with electron microscopy. You can see how the outer membrane has fewer proteins because it's less kind of bumpy to the eye. Now, the inner membrane, in particularly the inner face, has an incredible number of proteins. These are going to be transporters that we talk about today, but also think the complexes of the electron transport chain. Because this membrane is impermeable, small molecules and ions require proteins to transport them. That includes all of the small molecules we will talk about today. Before we get there, I'm going to make a quick aside to talk about NADH from glycolysis, because as it turns out, that is not transported into mitochondria.